Hello. Firstly, I'd like to thank the organisers for allowing me to speak at this meeting, and also to the previous speakers who have highlighted some aspects of 3D model adoption by industry that I will cover in my talk. My talk today will focus on the regulatory barriers to adoption of 3D models, and in particular microphysiological systems, in relation to how far we have come to closing the gap between technological developments and qualification for use in investigational new drug submissions by pharma. So in outline, I will give a short overview of some of the uses of MPS in pharma and summarise technology developments that have been made over the last 15 years. I will then focus the bulk of my talk on industry and regulatory adoption of MPS and highlight some aspects to consider for their use in non-regulated, investigational or clinical studies, concluding with whether we have managed to close the gap between developments and regulatory approval. Microphysiological systems, specifically in relation to the drug discovery process, have huge potential for use in a number of areas, including early in the exploratory biology phase, where disease models based on our scientific understanding of disease pathways may be created. These models are not subject to regulatory input, but may be submitted as part of an IND and be accepted by regulators based solely on their scientific validity. Those used in pharma for screening and internal decision-making, and increasingly for patient stratification and personalised medicine strategies, also do not receive regulatory input. However, when used for drug safety screening or to replace outdated legacy models of toxicity and efficacy, data and conclusions reached from MPS studies could be subject to regulatory scrutiny in applications for new investigational drugs with regulatory agencies such as the EMA and the FDA. In terms of technical progress in the MPS field, the US has benefited from pioneering technologies coming out of the Wyss Institute and MIT earlier this century, and subsequent funding by NIH and latterly DARPA to specifically develop a 10-organ chip as the goal of the tissue chip program. Since the start of the program in 2012 until late 2017, it was estimated that over 500 publications had been submitted to top-tier journals on the subject of MPS, and we have seen the uptake of those early technologies by commercial companies such as CM Bio for the MIT technology and Emulate for the Wyss technology. In Europe, similar programs of work were initiated as early as 2008 with the GoBio project, looking at developing a systemic solution for predicting long-term human substance exposure. Ultimately, this and other programs such as ORCID, the Horizon 2020 funded Organ on a Chip in Development program, comprising of these 7 main organisations in Europe, has resulted in models such as the PBPK-compliant 4-organ chip autologous IPS model for ADME profiling and tox testing. EUROX, the European Organ on a Chip Society, continues this work to promote adoption, and it has been estimated that R&D costs for pharmaceutical firms may be reduced by up to 26% by using organ on a chip technologies, representing around $5 to $7 billion in total. So technological progress over the last 15 to 20 years has been extensive, which is clear from the increasing number of peer-reviewed publications and patents generated. But challenges still exist, and we have heard about some of those in the talks at this conference. So how has this progress translated into adoption by industry and regulatory acceptance of microphysiological systems for drug development? Well, 4 recent publications from workshops held over the last 4 years between developers, end users, regulators and academia have really highlighted the issues and challenges to adoption of microphysiological systems by pharma and other industries, and have all made recommendations to reduce barriers to their use and application. In pharma, I mentioned the 3 areas where microphysiological systems may be used in exploratory work: preclinical safety testing and for patient screening and personalised medicine. Much of this has been driven by a not-for-profit organisation called IQ Pharma, and the group they have formed with major pharmaceutical companies such as AstraZeneca, GSK and Pfizer, called IQ MPS. This affiliate has has facilitated exchange of information between pharma and other stakeholders on the development and validation of MPS, and provided confidence to pharmaceutical companies to commit to these technologies. However, challenges for industry highlighted in the 4 reports still exist, and these are related to 1. The level of qualification required for a specific context of use, i.e. how and where the test may be used, and what specific data is needed by the regulators to approve that method for use. And 2, communication between all stakeholders involved in the development and approval of these methods. Repeated messages are that early engagement with end users and regulators is required to clarify their needs, and parameters to be measured and measures of success should be agreed early on. Sharing of success stories would also help drive acceptance. So the plan would be to move from a rather one-way street of communication from academia to MPS developer to end user to regulatory, to one where the developers interact with all stakeholders and there is a constant communication between all parties. For regulators, adoption of these technologies has provided a dilemma in the form of lack of experience with and confidence in these devices, because little data has been submitted by applicants who are not sure what data regulators want to see, but also because it may take 3 to 5 years before evaluation data is available through the IND submission. Up until recently, any data submitted would be looked at on a case-by-case basis, which meant that where qualification or indeed rejection has occurred, typically this information was not publicly available. So it is clear there is a need for regulators to indicate what data is needed, but also when an MPS method has been accepted. Suggestions for what regulators could do to help this process along are to publish a statement of need, establish performance criteria, and define a clear pathway for evaluation of new methods. So let's look at those in a bit more detail. Members of the Centre for Drug Evaluation and Research and the National Centre for Toxicological Research at the FDA recently published a paper on non-clinical safety testing strategies and the key needs for supporting human pharmaceutical development. This ranged from various aspects of safety pharmacology to general toxicological needs and specific toxicities such as assessing skin sensitization of mixtures, carcinogenicity, and development and reproductive toxicity, some of which could be served by MPS technologies. Performance criteria and measures of success have been initially suggested in EMA guidance for the introduction of new approach methodologies, primarily to address 3R's challenges in preclinical testing related to the EU Directive 2010/63. And amongst the tasks of the European Joint 3R's Working Group is the need to support qualification of these new methods. Likewise, the In Vitro Safety Working Group set up by the US FDA has development and evaluation MPSs as its first case study, and also has a performance criteria group specifically set up. Both continents have asked for clear processes and single entry points into regulatory agencies, and we have recently seen that the FDA has introduced a new webinar series where developers can submit information on their methods and platforms for review by FDA scientists. The FDA do already have medical devices and drug discovery tools qualification processes in place, traditionally set up for the qualification of biomarkers, for example. And these have increasingly been used for the qualification of new methods and technologies, such as stem cells. Crucially, both regions have testing centres, such as ECFAM, ICFAM, and the Tissue Chip Testing Centre, to independently test such methods. One example of qualification is from one of the Tissue Chip Testing Centres at Texas University, where a renal microphysiological system has been evaluated and results compared to the developer's data. In essence, transferability of the platform was successful, but reproducibility was greatly dependent on cell source, with commercially available cells performing better than primary cells. This study demonstrates the critical importance of developing microphysiological platforms using renewable cell sources, and in a similar vein, ICFAM are working to characterise the metabolic competence of some TOX21 cell lines and 3D HEPA-RG spheroids to support validation efforts in the near future. So some basic requirements for regulatory qualification of MPSs can be summarised as: the platform addresses an identified need and within a specific context of use. The method should be transferable between labs and the data should replicate published results. Beneath this are many other aspects that need to be considered, including harmonisation of cell sources, protocols and hardware, as well as quality control measures and correlation with relevant data, be it clinical or in vivo animal data. So finally, I would just like to touch on the challenges from use of microphysiological systems in the two other areas of drug development that I mentioned at the beginning of this talk, which are the investigational exploratory area and potentially the clinical area where patient-derived tissue may be used to determine the best treatment for an individual or cohort of patients. Here, standardization is key, and even before that, the vocabulary for standardization needs to be agreed so that everyone understands if we are comparing apples to apples or apples to oranges. Standardization needs to be community-driven and address common issues, and as for regulatory approved models, critical parameters must be defined and harmonized. Parameters such as dimensions, media, flow rates, and cell sources and handling are suggested in an ORCID white paper on this subject, and it is recommended that already existing guidelines be followed where appropriate, such as the Good Cell and Tissue Culture Practice version 2, a draft of which was released in July 2020 for stakeholder discussion, good in vitro method practices from the OECD, and recommendations from the International Society for Stem Cell Research, for example. Finally, in-house quality controls and qualification is required to understand how these systems can be used in a reproducible way that can measure functionality or perturbations of this functionality robustly. One area not explored in depth here is the interpretation and translatability of outputs from these systems, which, as we know, is already complex and likely to be more so as technology advances. While some microphysiological systems may still lack true systemic blood flow, hormone and immune systems, and many other defining in vivo contextual systems and cues, it is clear that developments in these areas are already making inroads, and it will be a requirement for more sophisticated analysis, such as quantitative systems pharmacology, to determine the true meaning of changes in vitro for the in vivo and population context. So in conclusion, the subject of this talk was whether we are closing the gap between regulatory acceptance and technological development of 3D systems such as MPS. And in reality, we know there are still many issues and challenges for all aspects of MPS development, whether for exploratory research, in-house decision-making, or for use in regulatory submissions for drug development. However, there are some common requirements which I think cross over all areas of use for MPS in biomedical research, and these underpin the regulatory specific requirements, which we have seen need to be addressed before these methods are accepted for use. These relate to standardization and harmonization of methods for industry, and can only be achieved through strong communication links between all parties in the development and use of these powerful technologies. What this means is that while we continue to be able to do more and more with microphysiological systems and achieve greater knowledge of the biology underlying disease and its treatments in this context, it is perhaps going to be a few years yet before we see firm ICH or OECD guidelines prescribing the use of this technology for drug development and screening. To get to that stage will also take a lot of interaction and collaboration between all stakeholders in the field, and it is clear that the process of adoption by regulatory agencies will no longer be linear and straightforward and will involve creative ways of working from all groups involved to achieve this success. Thank you.